All right, everybody. Good afternoon. Let me see if I can show my camera. Everybody could see my screen, right? I want you to see something. Oh, okay, good. Welcome to module one proper. Yesterday was introduction, and today we start the actual program. So I would advise you to start taking notes and start getting serious as from today because the work has started actually. So there's nothing holding us back anymore. So, what is bioinformatics, right? What everybody has said the word bioinformatics. By the way, you should know how to pronounce it well. That's why we wrote all of this. So, it is a branch of science that is concerned with information and information flow in biological systems with the use of computational methods in genetics and genomics. So the um the whole idea of bioinformatics is let's make sense of biological data because everywhere you generate data actually, no matter the field, finance, healthcare, agriculture, but bioinformatics here, we want to understand the information and information flow in biological systems in living cells, right? With computational method, right? So you 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 are here because you enjoy, let's say you have flair for biology. Of course, almost everybody here is in the biology, has taken a biology course one way or the other, and you have one or two interests in what's the name and in computation, right? So it's a good way of combining biology and computation. So the first term, the first definition by the term of mathematics was coined in 1970 by Pauline Hogewan and Ben Esper. By the way, I don't like history, by the way, but it's here, so we have to listen to this together. And when I ask you this question, don't expect me to be telling you when was the first 1970. You know, go sit down for my question. So that's by the way. Let's continue. So that is what bioinformatics is about. So that is by the definition of bioinformatics. People are still joining. All right. So it is a cross discipline. What I mean, it involves a lot of things together. A lot of fields are into play when it comes to bioinformatics. Provides CS with new challenges and clear medical significance. CS is computer science, right? So we are combining computer sciences and biology together, and of course mathematics. When you get there, there's a slide down that you'd see all of these other things that are involved. So it involves complex and large data sets, sometimes with very noisy and hidden structures. You would see that when we go along the way when you start seeing fast cues and seeing. You saw that first thing that's my opening slide that you saw a lot of lines. You would start seeing things like that, and your head will want to blow, but it won't blow. Don't worry, you would scale through. And can biological solutions be used to inspire new computational tools and methods? So biological part of the field, it enables you to analyze increasingly large and complex data sets generated in biological experiments. So you get data. By the way, okay, well, don't let me get tired of myself. So we sequence genomes every day. So before now, sequencing of genome was actually quite expensive. I think I'm getting ahead of myself, but it has become democratized at this point. So everywhere there is sequencer. The sequencer I have upstairs in my lab here is as tiny as, I don't know. I can. It's even smaller than my phone. Sequencer we have upstairs is smaller than my phone, right? But it does quite, like, it does what it does. So a lot of data has been generated. And imagine how many labs in Nigeria has that kind of sequencer. Imagine how many labs sequence every day. So you have a large amount of data. So we want to actually analyze those data and make sense of it. So bioinformatics facilitates the structuring and often unstructured heterogeneous data and to allow data integration for insights. Don't worry, this thing looks like jargon at the moment. You will understand what I mean over time. Just listen, pay attention to what I'm saying. So bioinformatics is that bridge, computer science and biology. So especially when realized in web service and tools. So very soon you will start seeing, you should have seen all of those movies where when you are where you would see somebody hacking, planning an hacker using blue screens and all those things. You're about to enter that realm, by the way. So well, of course, you're working in biology because if somebody asks you to build the software, you might not be able to build them, but you can still use the same programming language that you're using to build the software to also work with. You are using the tool they are using to build the software to actually what's the name, to analyze biological information. So that's why mathematics is cross-disciplinary. And people that build, I also mentioned mathematics, by the way, because people that build the tools we use also, some of them are mathematicians. They also understand biology, but they are mathematicians. So it's very important for you to know that this is not just a one man's food, it is cross-disciplinary. So I won't be surprised if in our next course we saw mathematicians in this training, hoping to join us and learning from us. So that is that. Next slide. So saying again about mathematics, so it sits in the middle of biology, medicine, math and physics, and computer science. So everybody comes together to make the tools, to analyze the tools. Some of the algorithms we use are old and very, very complicated mathematical algorithms. You'll be surprised when you start understanding. Even maybe if you are here, if you might have understood, you might know. Was I lagging? Can you hear me, everybody? Okay, good. So I was saying earlier that you that's keep my mind. So my mathematics sits admits all of these things: biology, medicine, mathematics and physics, computer science. It sits on top of all of them. I was saying earlier, okay, the algorithm, one of the algorithms we use sometimes, if as simple as blast, if you have heard of blasting or NCBI on it, all of these things before, the algorithms that sit underneath blast are crazy, crazy mathematical algorithms, right? So made sure that means a mathematician somehow or somebody that studied biology had gone through and crossed to mathematics to see that okay, this algorithm will be able to help us solve this problem, and they inculcated it, right? So if you, I hope you take this work serious more than what we are doing here, and you move beyond past just using tools to analyze and you start building tools yourself, you discover that you would have to actually go into mathematics and learn one or two things together to understand one of these things. So bioinformatics has all of these fields in together. So biology, medicine, maths and physics, computer science, that is what brings you, that is what we hook up together to get that general term that we call bioinformatics, right? So you would start asking me, will I write mathematics? No, don't worry, you will not do mathematics, by the way. You will not do it. Will you do it? No, you wouldn't. So you don't have to fear for people that are already scared of mathematics. So please, you don't fear. Right. So the bioinformatics enigma. So let's dispel the truth, the myths, and reveal the truth. Let's. So this is a quote by Richard Dobbin. I do not think of biology computing is mathematics. Example, mathematical modeling is not bioinformatics. I know someone that does math that is interested in mathematical modeling. It's not bioinformatics, it's something else. Even when connected with biology related problems, it is not. So in my opinion, bioinformatics has to do with management and the subsequent use of biological information, particularly genetic information. So anything that helps you or that involves you using biological information to do work, right? That is bioinformatics. You don't have to all this what's the name? The mathematical modeling, this, that, you don't have to, it's not part of, it's not that's why I'm saying you don't need mathematics, by the way, except you are interested. Except you are interested in, by the way, in mathematical modeling specifically for health service, right? But if not, please, you are very, very much free to ignore those parts, right? Mathematics has nothing to do with mathematical modeling at all, please. So what does bioinformatics involves? It involves the use of genetic material, DNA, RNA, what's the name, proteins, right? Transcriptome, different type of things that are in that central dogma. You know the central dogma of life from DNA to RNA to protein. So every process that is involved in DNA, RNA, protein, and computation around revolving that is what is called bioinformatics. Nothing else outside that, right? So you see people working in transcriptomics, people in working with protein analysis, different things, by the way. So we are going to be teaching you based on genetic information. So DNA majorly, and then some RNA. Proteins too. We'll move into protein. We'll talk about it, double to it once or one or two times. But we're not going to be talking about the transcriptomics and all the likes, because if you want to, what we teach you is enough for you to pick on that if you want to actually go ahead and do transcriptomics and all these RNA-seq and chip-seq analysis. right? So, um, the myth, what is not bioinformatics? So people say bioinformatics is just biology with computers. It's not just it's not merely plugging biological data into spreadsheets. We are not even going to use spreadsheets. Who uses spreadsheets, by the way? When there's Hawk and grep, be Hawk and said all these commands, we don't use spreadsheets. So it's not just using computer. And if you think you're using spreadsheet to import, it's not even efficient. So you don't have that. And then not no simple data entry or basic statistics. Bioinformatics demands algorithmic innovations. I told you earlier, the algorithms we use to that powers the tools we are going to be using. Some of the tools you will start using maybe from next week, from next 2 weeks, you would see that you just sometimes I imagine, by the way, how are these tools built? And then you see if at the root and the core of all of these things is mathematics and physics, like maths and physics, hardcore maths, hardcore physics, right? So I think it is very, very crude for you to say mathematics is just biology with computers, right? It's not just biology with computers. Myth number two, is a pseudo scientist. So, uh, you, how am I going to explain this? It's not just somebody that is sitting down somewhere crunching numbers. If you see how mathematics is, you know it is before even before the data gets to you, a lot of people have worked on the data. Somebody cultured the sample on the bench, somebody extracted the DNA, somebody prepared the library, somebody sequenced it, and then the data is out for you to work on. So you are not just a sole data scientist working on it. So it is like it's bioinformatics thrives on collaboration, global teams, diverse data sets. There are data that want that I've seen teams that they don't have just one bioinformatics, they are bioinformatician. They have like 5 to 10 bioinformaticians actively working every day on a large data set. If when they invite if you finish here and you get a job, and it's not just a small team, it's a large team, and you see data set and they ask you to analyze, you said will fear. You would fear. So you know that sometimes you have to pick up. This is not just a one-man job. This is a team's job. And of course, you collaborate internationally on different projects. Very soon you start doing that. So the fact that you are saying solo, it's not a solo scientist for war. That is not what this is, right? So it's only about DNA sequencing. See, I said it earlier, it's not limited to say DNA sequencing, right? And genome, it transcends raw sequences. It's about interpreting data from medicine, agriculture, and beyond. So agriculturists too. You see, most of us buy agbado, maize, corn, whatever you want to call it, and they culture it in 2 months. It's out, and you discover that the wild type maize that we know will take 3 months, 4 months, 5 months sometimes before it shoots up of the ground and becomes edible. And we have coconuts that they are not as tall as they used to be. These things are genetically edited, and before they could genetically edit it, they have sequenced the DNA of coconut, and then they have troubleshooted with bioinformatics analysis to know which of the genes is responsible for the height. And they found out this is the gene. Okay, we can tweak this gene so that it does not, or we can knock out this gene so that it does not go distorted. So this is the gene responsible for the height, right? That's agriculture. So they will make sure, okay, if this gene is responsible for the height, let's remove it. Then the organism, the plant will not be this tall anymore. It's just going to be this high, right? And this is how, so that anybody can come in and pick it. And you see some sweets or some coconuts or some whatever palm tree that has very nice, or mango tree that tastes very nice. Of course, we have or people here would know seedless watermelon. There are watermelon that are seedless, no seed at all. So how did they get to that end point of discovering a seedless watermelon? It is a lot of genetic analysis and bioinformatics, right? So they sequenced a wild type, what's the name? A wild type mango or a wild type watermelon. And then after they grew and sequenced the organism, then they went to bioinformatics and did a lot of analysis to see, okay, these are genes that are responsible for the sweetness. These are the genes responsible for the eyes. These are the genes responsible for the color. So if I want a what's the name, a mango or a watermelon without the seeds. So these seeds are going to be terminated. So they knock out the gene that is responsible for the genes, what's the name, for the seeds, and then they put that back in a living what's the name, a living watermelon, and then it grows from the ground, and then you discover that, oh, we have a watermelon that doesn't have seeds. That's very, very much common at these days, but maybe not in Nigeria yet. But in the Western world, you see seedless watermelons. You see seedless mango, a whole mango that when you open it, you wouldn't see the hard seed inside of it. So before they get to that stage, that's an application that you can see physically in agriculture, right? It has gone through a lot of bioinformatics behind, right? A lot of analysis, a lot of wet lab experiments, by the way, because you can't do bioinformatics alone. To validate your experiments, you have to actually do raw wet lab sciences, right? So these things go hand in hand. So you see me talk down the line that mathematics is not just solo man. It's you work in a team. You don't just work alone as a mathematician. You work in a team. You work with researchers facility that are doing one or two things, right? Then um the next question is, what else is not mathematics? Biologically inspired computation, e.g., genetic algorithms and neural networks. Then however, the application of neural network to solve some biological problems could be called mathematics, right? So now we are in the we're in the world of what's the name? AI and neural networks and all of those things. So not all of them are actually mathematics, so you don't have to get scared. But there are some analysis that you would see maybe they want to do some very, very crazy analysis. Let's say pan-genome analysis, GWAS analysis, quite a number of things you want to do that might be very, very slow, and you want to use neural network to help you classify and do one or two things. So you are doing bioinformatics, but you are just applying the tool to help you. You are not just sitting on neural network and think you are doing bioinformatics. So when it depends on the question you want to answer, by the way. Bioinformatics is answering questions. So what's the question you want to answer? We data will help you answer the question. The genetic data, most of the case. After you've gotten your data, then you go ahead and answer the question, right? With bioinformatics. So what really is bioinformatics, right? It's a fusion of biology, computer science, and statistics to decode life's complexity. I talked to you about it already. Biology, you have to know genetics of the organism you are dealing with. Then computer science to be able to help you pass through the data you want to pass through. And then statistics, you want statistics to be able to decode life science, right? That's basically what it's about, right? So statistics is not invariably there, but statistics helps you with a level of confidence for your results, right? So you will start doing statistics from the first or second week, not the statistics you do. Don't be traumatized. For people that are traumatized with statistics, me too, I am. But you would understand what I mean when I'm saying that, okay, the accuracy of my fast skill raw reads is 1 in 99%, or the probability of an error is 1 in 1 million, or 1 in 1,000, 1 in 100. You would know these things over time, by the way. So that's like an element of statistics. Even some when you do blasting to when you use some algorithms, there's an element of statistics that is involved in all of these things. So you are using biology through you are using you are working with biology through computer science and confirming your confidence with statistics. So, and as of 2025, it powers AI-driven drug discovery and genomic breakthrough. Some of us have heard of AlphaFold. I hope we have, in proteomics. Google. This is not even new. I think since 2010 or 2011. I don't know. And they were able to use what's called AI to actually predict protein structures. So if you have not known it, just Google AlphaFold and read paper, any paper that comes on AlphaFold, you understand it. Before they got to that level, a lot of analysis mathematically have been done, right? So even those discovery till now, say, new drug discovery processes are gone through, like AI has powered it. So bioinformatics, before they get to the AI part, they have done quite a number of bioinformatics. So they just use that. So as I said earlier, they use that to predict. So um bioinformatics is basically fusion of biology, computer science, and statistics to decode the complexity of life, right? So also it is it is a process of moving from data to discovery. So bioinformaticians collect a vast amount of data. You start seeing itself. Maybe when to start with, we'll give you little data because most of us are using computer with 256 gigabytes ROM, and sometimes some people have 128 gig Chromebook. Some people, by the way, don't bring Chromebook here. So some people have what's the name? What would I say? Very slow. So we don't want to give you RAM size, by the way, because you don't want to load in the amount of if you load much amount of data to your RAM and then your computer crashes. So we don't want that. So, but bioinformatically, when you start working with actual real data sets, you would work with vast, like in gigabytes and terabytes of data to do analysis. Particularly if you want to do world-class analysis, you would work with a huge amount of data. And then you'd analyze this data with advanced bioinformatics algorithms and machine learning processes. So different algorithms have been written, told you mathematical aspect. They are written by algorithms for themselves, but we discovered that we could apply those algorithms that are written to our own analysis. Then we inculcated it, so we just adopted the data, the algorithm they have. So we analyze that data with the algorithms mathematicians have used, and then the results we get translates into actionable insights for science and society. Sciences will tell you when of course you were there where prof were teaching open reading frame and all of those things. You will be surprised how okay bioinformatics help us know this is an open reading frame. If this is an open reading frame, then they might be the may or may not be a gene. So how do I confirm that there is this gene is responsible for something? You can do PCR, you can do a knocking out experiment, you knock out the gene, see what is affected in the organism. So you can see the whole process from start to finish. Data must be gotten first, then after the data is gotten, you work on it with mathematical algorithms or mathematics machine learning algorithms, and then after that, you go ahead to pick actions, take actions to work on the data, right? To work on actions that you have to inform actions, inform responsible for be what I would say, take insights from the results you got through your analysis, right? So, and what's the impact that it has done in 2025? It drives precision medicine, infectious disease research, and pandemic responses. So, um, everybody was aware when 2020 was the pandemic hit, right? So, if you notice, one of the countries that is the first responders, or that have, I think they have the highest number of genome sequenced globally, is the UK. And you'll be surprised how were they able to get that large amount of genomes analyzed and number one sequenced, then number two analyzed in real time. So at the point they were generating millions and millions of reads every, I think 8 to 10,000 every reads every data every week during the pandemic period. How were they able to cope with this huge amount of data? How were they able to get all of this processed in real time? So it turns out that they have infrastructures in place, they have bioinformaticians in place, they have quite a number of things. Mathematics analysis, because of course everybody knew today you see there is Omicron variant, tomorrow there is Delta variant. Those names we are getting are from bioinformaticians. They are the one getting the names out. They are the one doing the lineage assignment to the reads that sequencers have done. And those things you're getting are actually real-time bioinformaticians telling you, okay, now we have a new variant that's different from the variants we have before. And okay, this is VCF, very simple, they did the variant calling file, all of this, but you understand them over time, don't worry. So it drives, and because there's a new variant, right? They know okay, the drugs we used before might not work now. The drugs you use now might not. We might have to get a new target. So they will just look at the genes and the features of the organism bioinformatically and say, okay, we can draw. We work with a pharmacist or informatical dynamic person that would help us to. These genes have this very place. We can make a drug that will target this place, right? So if we have a drug that can target that, that's you will be so precise with the drug. You know when you put the drug inside, what's the name? When you put the what's the name? The drug with the organism, you know that it's going to work, it's going to kill it, or it's going to destroy it, whatever you want to make the drug do. Precision medicine, whatever you want to do, you can alter it from the genetic level. And the only way you can access the genetic level is through bioinformatics, right? And of course, it's a global force empowering LMIC researchers with advanced and innovative tools. So me and you, we are from LMIC. LMIC is low, middle income countries, right? So low and middle income countries. So this has really, the impact of mathematics is that help us to do quite interesting researches, right? We might not have the wet lab experience that the Westerners have because they have gone far ahead. The things we are doing now, they have done it maybe 10, 12 years ago. They have moved ahead of us in the wet lab. But in dry lab, when it comes to mathematics, we're at par. Like I would say that we're at par because what they do, we can also learn, we can inculcate. And of course, technology is democratized. They have GPUs, we have GPUs. They have clusters, we have clusters. They have science. The only thing we need to do is learn. And that's why you are here, right? So to learn and be able to do crazy, crazy things with mathematics over time. So why is mathematics necessary? So the need for mathematics has arisen from the recent explosions in public available genomic information, such as resulting from the Human Genome Project. So we I think the first Human Genome Project was published in the year 2000, and it was edited, of course, 2003. Now, it was very, very hard when they were sequencing the human genome, because if you know the process behind it, it was humongous, it was serious, but they did it, right? So at that time, very few people were sequencing globally. Very limited amount of people were sequencing globally. Maybe four, six, but I can move here. So very few people were sequencing globally. And what has happened is COVID. When COVID came, sequencers became democratized. Everybody has sequencers, because we saw that to be able to beat this pandemic, we have to know which clade or which lineage or which variant is currently floating where or when. So to make this happen, everybody started having, the NGS now, next generation sequencers started coming out everywhere. So today it is Illumina. People started having Illumina everywhere. Illumina, of course, Illumina is bulky, but then Nanopore came around. Nanopore sequencers, very small, very minute. Of course, there are bigger ones too, but the fact that Nanopore came and they had this MK1B ones that you could use, very, very plenty, very small sequencers. So people would sequence in Lalukmo or from Bauchi or from what's the name from Maiduguri, Borno State. You can sequence from anywhere, even Congo. War-torn areas you can sequence, because um there was democratization of data, right, of sequencers. So, and the cost of actually sequencing was dropped a lot. So why is bioinformatics necessary? I think I digressed a little bit. We have a huge amount of data now, right? There is a lot of publicly available information. We have a lot of databases on the internet, NCBI, ENA, SRA, different archives of data. And then we need it to gain a better understanding of gene analysis and taxonomy and evolution. So we have to understand and keep track of our organisms, of ourselves. So all of this are put in place and are necessary for us to be able to, are the reasons why bioinformatics is necessary. That's why people like us are actually needed in this field, right? And to work efficiently on the rational drug designs and reduce the time taken for development of drug manually. So before all of this mathematics came, the way at which they test and discover drugs was archaic and long, right? But now we have different tools that could help you hasten and reduce the time of from inception to drug discovery. For the whole drug discovery pipeline has been shortened because of bioinformatics. So we are actually needed here in like we are needed as much as possible in working in global rescue. Let me use let me use that word. So um human genome project and next generation sequencing, I think I mentioned this earlier, but this is a visual explanation to what I just mentioned earlier, right? So human genome project was published at the year 2000, was first published in 2000, but was perfected at 2003. I said that earlier. And what they did is they want to know the human genome. They want to sequence it from beginning till the end, right? And it cost a lot of money. Like it was humongous. Like it was pretty, pretty huge. It was expensive. And there's this COVID came, right? Next generation sequencing became introduced by the way. I think from 2006 to 2020, a lot of this technology was invented. We have Ion Torrent, we have PacBio, we have Illumina. These are the major players in sequencing. So if you don't know those, you can actually write them and go and read about them. PacBio, Ion Torrent, Illumina, then Nanopore came around later in the century, in the decade, right? So this next generation sequencers were introduced, and then they crashed the cost of sequencing per genome. So you can see earlier we have about 100 million to sequence a human genome, like $100 million to sequence just one person's genome. But you can see it now at the current rate is just a little above $1,000 per person, right, per genome. So it has crashed a lot because of, I told you earlier, because we have sequencers that are cheap, we have mass produced sequencers, and we have also increased the capacity of scientists. We have trained our scientists to be able to actually do quite a lot of things. So over time, we have crashed the price of sequencing, and now everybody has data everywhere. So there is enough data for you to practice and work with. We just have to sit together, learn these things, and then start doing cool projects globally, right? And I said genomic data boom. I think I mentioned it earlier. I think if I'm repeating something, we go there, we go listen to them together. So just wait and listen to me. So you can see that in year 2015, we have just 13,572 genomes. 2016 increased to 17,000, and 2017, 25,000, 2019, 47,000, 2022, 72,000. Like the genomes keeps increasing. So people putting data on this our network archive, our data archives globally, NCBI, ERA, ENBL, different places, DDBJ. So data side, everybody started submitting data everywhere, and then we have a lot of data now, right? GenBank database, you can see basis per million, quite a number of data. So see GenBank database, see how it moved up. Sequence read archives basis, you see how much it went up. And what's the name? SRA open access database. This chart, this slide has shown you different data. You see how much they have scaled up and increased over time. So SRA archive databases increases. SRA, by the way, are short read archives. And when you hear short read, they are talking about technology like Illumina. So Illumina became very popular. People started using and sequencing. One of the biggest breakthrough in this of the century, by the way. So over time, next generation sequencing has empowered us to be able to sequence and put up a lot of data. So a lot of data has been in place. And one thing is people share actually, except for some, there's in cases that when there's I'm not sure if we have a section of data sharing policy and all the likes. I think we should also later. But um a lot of people start sharing data, like sharing a lot of data. It is public database. When you are done with your data, publish it on NCBI, publish it on DDBJ, SRA. There's this one that we use in the lab. I can't remember the name for now. So we post just people are posting data all around the globe. So there's no hoarding of data. Nobody hoards data. We just share, and then we have a lot of genomic data to use, right? And now that we have this genomic data to use, we have what's the name? We have a lot of data to play with. So all we need to just play with this data is the computational aspect of it. So you need actually to work with this amount of data, you need a very strong computer. And most of these data centers actually have clusters that you could use to, and you can run analysis on them too. So that's not even a problem. And DNA sequences, we're heading somewhere here. DNA sequences are millionaires. Of course, I told you earlier that people started putting DNA on different servers, right? You can see the amount of DNA sequences that people have pasted online. But these reads alone are just meaningless. You can just stare at this all day. You don't understand what it is. You don't know what it is. What do you mean? You can just ATGC, ATGC, TTTTA, whatever. But making meaning of this data is now what we are here to do. It's what you'll be doing in the next 4 to 6 months. I hope it's not more than that. I pray it's not more than that. So 4 to 6 months maximum, you would understand how to navigate your way through this data, right? So DNA sequences are minimum. Yeah, it's meaningless. Then what you want to do, what you'll be learning in the next 4 to 6 months, is how to uncover the wealth of biological information hidden in the mass of sequence, structures, and literature of biological data. So you would be surprised to see how many things are hidden in this data. This data that you just saw here, a lot of things are hidden here, right? And it is being used for in now and in the foreseeable future in areas of molecular medicine, different areas of molecular medicine. I'm trying to remember one scenario. Okay, let me paint an example. I think there was a patient that had an MDR. So they isolated an organism from the patient in the lab and they discovered that it was MDR. They tried a number of antibiotics, and of course, if you're a scientist, you know there is no zone of inhibition, no organism. But I think what happened was they sent the sample to the lab, a research lab, I think UI, and they sequenced it. and they use bioinformatics to check for other resistant genes. So there is a AMR database. So if they discover that there are not all the AMR genes are present, so they will use the drug that does not have the AMR gene in the patient. I hope you understand. So it has helped in what's the name in um nuclear medicine. I'm sorry, just say nuclear medicine, in molecular medicine, to be able to help patients, help isolate, help. A lot of things have been, and also in molecular medicine, it's also used in environment in identifying waste and clean up bacteria, in identifying waste and cleaning up and dealing with bacteria. There are some bacteria now that I think they are proposing that could help um, clean up oil spillage, right? So these bacteria have been seen to actually degrade oil. So why not we just engineer the mechanism that they use into another organism or something so that we can mass produce this and clean up oil spillages in places like Niger Delta or Sudan or wherever the oil spillage things are happening. So this are actually the goals of bioinformatician. So we have the organism that has the potential of cleaning up. Okay, there's a gene. Let's isolate that gene. Let's see how we can modify the gene, then put it back in an organism, and then see how much work it does. So you can imagine how much application is going on. Different process you see, by the way, in biology has bioinformatics. One crucial part is bioinformatics, right? This is very, very crucial in there. And then in agriculture, I explained what we have done with bioinformatics in agriculture to you earlier. So high yield, low maintenance crops that can resist drought, crops that can resist what's the name? Um different harsh conditions. You will see crops that they would cactus, different things that would in very, very so what they do is they would, let's say, cactus can stay up in a arid or very dry desert, and they would explore sequence the cactus, look out for the genes that are responsible for their toughness and ruggedity, how they are rugged, what is the gene aspect that is responsible for that. Then they will cut out that gene, then put it in an organism that is not able to survive in an environment like that. And then you see the organism that is not able to survive now becoming very, very strong and very resilient to harsh weather conditions. So, excuse me, bioinformatics has helped a lot and will help. And these are the goals of bioinformatics going down. So while we are going to be teaching you, we'll be teaching you most of the majorly microbial genomics, right? This is one of the simpler paths to enter into bioinformatics to understand. If you want to do proteomics later, it is up to you. We'll teach you everything that will be in the foundation for you to learn better, right? So transcriptomics, proteomics, it is up to you. We are coming in with basic microbial genomics, and then you would understand that, and then you can move on from there. So these are the goals of bioinformatics. Then we can go ahead to the next slide. Okay, I think it might sound like repetition, but you will hear it with me. Where bioinformatics help in experimental molecular biology, in genetics and genomics, in generating biological data sets, analysis of genes and protein expression, and comparison of genomic data, and simulation and modeling DNA of RNA and protein. So a lot of things and a lot of usage of microbial genomics. I'm trying to remember an example. So they are trying to see if a drug can help you kill something or something. So they will set up two experiments. One organism is exposed to a drug, one organism is not exposed to a drug. And then at the end of everything, they would, at the end of the experiment or at the end of exposure, let's say they expose them to a drug for a week or for 2 days or 3 days. Then after that, they would extract the DNA of the organisms, then sequence the DNAs or sequence the RNAs, depending, and then they can check the rate of expression. Mathematics will help you to now, okay, now that I've gotten the DNA, what genes were activated, what gene were pressed, different things. You would qPCR could help you with a part of it. Then if you want to go deeper to do higher resolution stuff, then you now start working with bioinformatics to help you understand these things. Different things, different. By the way, I should mention this. Here's somebody say I'm a bioinformatician in the US, in the UK, you should be sure what bioinformaticians they are, because some people are working in human genomics. I am a microbial genetics bioinformatician. I don't know anything about human bioinformatics. So if I want to pick it today, I would get up, I would move on fast. But another person in human genomics bioinformatics might not know what we do in microbial genomics. So you should put that in mind. Even aside that, Somebody is strictly a bioinformatician in transcriptomics. That's all they do. They don't know anything about our own part. So they might have an idea of what we do, but they won't be able to get to our level of expertise. So you should know that there are different parts of this bioinformatics. I think I mentioned it earlier. So we'll teach you through bioinformatics and my microbial genomics to come in. Then over time, you can now start picking through whatever part you want to pick on, right? So what are the branches of bioinformatics? There is statistics and machine learning. So mathematical and statistical modeling. So you would be confused, or you might be confused. I mentioned that mathematical modeling, it's not bioinformatics. There is a part of bioinformatics that you borrow mathematical modeling, but you don't just sit down doing mathematical modeling for biology and not include genetics and call that mathematics. They are two different things. Then computational biology. So here you're working with HPCs, you're working with database softwares and algorithm development and web services. By the way, sorry, most of us here are from Nigeria. I think everybody is from Nigeria, and everybody here is from UI Ibadan. Sorry, your school doesn't have an HPC. Western schools have HPCs, and I think that's one of the reasons why they had moved faster in what they do. I didn't even know about HPC until when? Maybe until I was doing my NYSC. The same time I discovered bioinformatics. So when I saw that undergraduates as early as 100 level, 200 level are using HPC clusters for the analysis, I was impressed, right? Because this is what PhD people are doing here, and I still paying through their nose to get used to, right? So that's just by the way, by the way. So classical for bioinformatics, that's where you do things about structural bioinformatics, gene prediction. You want to know what you want to predict what the gene is doing, motif prediction and pattern matching, gene expression analysis. I mentioned something about gene expression analysis earlier, genome annotation, and the molecular phylogenetics. Sweet parts of I think we are going to cover a lot of classical bioinformatics here. Also, one thing that bioinformatics does is system bioinformatics, system biology, network biology, drug design, image analysis. Mathematics is scattered across all of these things. We are not teaching you everything, no. So, but we're going to help you understand. And if you want to move on to all these things, you will definitely understand and know how to navigate your way. And then statistical genetics, genome-wide linkage and association study, GWAS, beautiful stuff. It may be complicated a little bit, but you would understand it. Don't worry. Genotype analysis, population genetics. So these are different branches of bioinformatics. Everything we will shall start time next week. So if you are sleeping, because I know yesterday they said people used to sleep, don't sleep in this class. So maybe I'll ask you a question next week. Maybe that's why I'll start kicking people out. All right, that's just by the way, don't mind me. We mentioned genome annotation earlier. What is genome annotation? It is genomes are marked to know their regulatory sequence and protein coding. So it is very important. It's a very important part of Human Genome Project, and it determines regulatory proteins. So when we sequence a genome, like we sequenced earlier that I showed you, that ATCC, that slide I showed you, right? The what genome annotation does is when you have the whole read, you now want to start compartmentalize. You know, there's a difference between gene and genome, by the way. Genome is everything, gene is individual parts. So you want to know what gene is responsible for what will I call it? What gene is responsible for your skin color? What gene is responsible for your hair color? What gene is responsible for your height? Or people that are short, sorry. What gene is responsible for your what's the name? Intellect, right? And people are smart by default. They have the chance to be smart, by the way, because of the way some of their genomics are. What gene is responsible for your physique? Some people have rounded bodies, some people have boxy shapes of bodies, some people have wide. So genome annotation is where after I sequence the whole gene, I want to know, I want to now name each gene. So this gene is responsible for my eyes, this is responsible for my nose wideness, this is responsible for my nail color, this is responsible for my hair color, like different things. That's what gene annotation is simply, right? So the whole process comes from after you get your DNA, the whole DNA sample, you fragment it and you want to know what each of the patches. So this is a gene, there's gene, what's the name? There's a gene, there's a non-coding region, there's another gene, then there's a no-coding, there's a repeat. So you want to, the old gene, you want to understand what each part of it does. So between genes, it might be a very silent part where it's just repetition and nothing. And then you see another gene, sometimes you see 3, 4 genes overlapping each other. Of course, you guys have done ORF, so you understand what I'm talking about. Different reading frames, different genes, alternative splicing. So All of these things fall under genome annotation. You do that over time. We are going to teach you genome annotation by the way. And comparative genomics. So comparative genomics determines the genomic structure and relation, functional relation between different species. So if there's a gene, let me read the whole thing and then explain. So for this purpose, intergenomic maps are constructed, which enable scientists to trace the process of evolution that occurs in genomes and different species. So we all were scientists say we all were a ball of gas and then they explode and then everybody evolved over time, millions of years. Some people choose to evolve to dogs, some people choose to evolve to human beings. A lot of things happens, but there are some genes that were that express different way in all of them, right? In all of us. So there is a gene that's responsible for our hair color, like the color of my ear or the color of my skin. And the same gene is responsible for the color of the hair of a dog, right? So different species have different genes manifesting in different ways, like different the same genes rather manifesting in different ways. So comparative genomics, it is very used in evolution, by the way. It is used more in evolution to know the process of evolution. That's when you start talking about homology, autology. All of these terms might be a little big, but over time you would understand as we move on, right? So this thing is it might be look, don't worry, you will understand it over time. So um taxonomy of different organisms and then the genes that are overlapping in each of them that somewhat does the same thing or not. Also, I said prediction of protein structure. So it's easy to determine the protein structure of proteins in the form of amino acids. I think I mentioned something about alpha folding before, right? In the form of amino acids, which are present on the DNA molecule, but it is difficult to determine the secondary, tertiary, and quaternary structures in protein. So tools of mathematics can be used to determine the complex protein structure. I jumped ahead of myself in the beginning of the class. I talked about alpha folding, right? So mathematics was actually used heavily. Mathematics then AI was used heavily to predict a lot. So if you have, I think if you have any sequence now, if you have just the sequence, the FASTA file, right, you can predict the whole protein folding protein structure with AlphaFold, and that's because of bioinformatics, right? Bioinformatics on steroids. So we can use bioinformatics. It's one of the goals to use to predict protein structure. Very beautiful thing. And if you don't understand what protein structure is, I think you should read a little about that. Protein is from alpha helix, then to beta folding, and then they have different structures, structural level from the primary to tertiary and then quaternary structures of proteins. So it's a lot of scaffold upon each other. That's what makes protein does what they do, right? The way protein, I think almost about everybody, well, everything in our body is protein, by the way. So moving on to the next, of course, why am I even saying that? We talked about central dogma from our DNA to RNA to protein. Protein is the end point of everything. So everything in your body is actually protein. Excuse me. Then health and drug discovery. Say I don't repeat them, Abi. We go here and together. The tool of this mathematics are also helpful in drug discovery, diagnosis, and disease treatment. Next generation sequencing of whole organisms have enabled the scientists to make medicine and drugs, which can target various genes. So let me tell you a little scenario. The fact that I've annotated a gene and I know what each gene does, right? I can kill that organism or enhance that organism because of that, right? So if I know this gene is responsible for how rugged you are. So if I know there are some organisms that if you kill them from now, see, if you put them in hot water, there are some spores, right? If you even that's why you have to autoclave at 121 degrees for 15 minutes. If you autoclave at 120 degrees, they still no go die, right? They are rugged, they would live. After you have done that your autoclaving, they will tell you your day shit, they will still live, right? So imagine I know the gene responsible for their tenacity, then I can get that gene and target a drug that would kill that gene so that if any human being have or inhales that spore and that spore is affecting them, once they take the drug that I know that can target that gene, then the person will live, right? The person would not be, they already understand that's the whole process of drug discovery. So a lot of genome annotation, and then once you know the work of each of the genes, then you can start building drugs. Even I think new part of AMR discovery now is AMR, because we know a lot of AMR and antimicrobial resistance is happening globally. People are, that's the new pandemic, by the way. We just don't know it yet. So a lot of people are taking drugs that they're not supposed to use, and it is causing, or it is fostering resistance. So if we know the resistance, of course we know the resistance, most of the resistant genes, we can build drugs that will target those resistant genes, so that new drugs, by the way, new antibiotics, that will target those antibiotics and those resistant genes, and then the organisms will become the organisms will die, and then we'll live our life normally again. So we can see how bioinformatics is helping us identify drug discovery and doing this. There's a whole process, but biological data to computational analysis, then identification of target. I've explained all of those in this my small story. And then drug design and screening, so molecular drugging, docking, virtual screening, and then they will test on the bench, experimental validation, and then they can now start giving personalized medicine to individual patients, right? So beautiful, beautiful stuff. Preventative medicine. So gene identification by sequence inspection and prediction of splice sites allows mutation to be corrected easily. For example, this is much used in analysis of mutations that causes cancer. So if they know the mutation that can cause, I think there was an experiment that they did for this is not even cancer, this is sickle cell. Of course, we know the mutation that causes sickle cell, right? So what they did was they used CRISPR to to please hold on. So yeah, thank you. I'm back. So preventative medicine. So I was talking about genomic. I was talking about what's the name? The sickle cell. So what they did was they built. Of course, they know of, and bioinformatics have helped us know that I think valine changed to glucamic acid, right? So they went to genetic basis of the valine, the codons are responsible for valine and glutamic acid. Then they built CRISPR targets. Of course, you will know CRISPR. I think we should know CRISPR, by the way. It is popular, and everybody on this call should know what CRISPR is. So they built CRISPR targets that would excise that point, and then they would repair that part back, and then they give it to the patient. So the before those experiments are done ex vivo, like in vitro, so outside the patient. But there was an experiment that was done, I think last month or 2 months ago, and they created the CRISPR targets, they created the repair mechanism, they made it into a pill, and they gave it to the patient, and the patient recovered. I think the patient is still they are still monitoring the patient, but the patient recovered. And the sickle cell gene, I think they have the sickle cell, most of the gene has been repaired, by the way. So you can imagine how we can use this going forward. So there are parts of the world that before your baby is born, they can actually do a panel screening to see what are from the baby's genomics. So they would take a part of genomics and then see what kind of disease is this baby likely or susceptible to, likely to get when it comes in life. So they know, they will give preventative medicine. So let's give this injection, let's give this, and let's give that. Then it will help the baby to be fortified against this disease when they come back to life, when they come to life, or when they come out of their womb, so whatever. So this is very important. Then, of course, in cancer, when they know the genetics that is causing the mutation, they give you something that would repair it, right? So from genomic analysis, from data integration and standardization, genomic analysis or epidemiological analysis, data integration, they will understand pathogen surveillance, host pathogen interactions, drug discovery and resistance. A lot of things to be done, right? So public health interventions and surveillance. So it is very important. I currently work in wastewater epidemiology, and you would be surprised to see that when we work, we go to wastewater, that your dung, that your rivers and different places that you people dump your water refuse, because I know you Ibadan people are very, very dirty. We get the water from that place, and then we concentrate in the laboratory, we look out for pathogen. You will discover some places that they think they are fine, they are not fine. You see how much Vibrio cholerae we got from their water, how much Salmonella typhi we get from their water. And then after we have used what's in qPCR to find this out, then we go ahead and do amplicon sequencing. Then you now see the actually the genotype of the DNA that is living in that environment. Some 2.3.1 clade, 3.1.1 clade. Like, it is very important, it's very interesting. You see Also, we do clinical surveillance at the same time. You'll see the clinical surveillance part of it when the culture and then we get the organisms, we go ahead and sequence it. We see clade. You would be impressed to see that the clade that is circulating in the environment is also mirrored with what is circulating in the clinic. So is your people that is actually going out to the wastewater that we can see and pick out. So you can imagine and you can see that you can do a lot of public health surveillance that way. Because not everybody that is sick comes to the hospital. But everybody that is sick, we pour water into the wastewater or we shit and then their shit will go into the wastewater. So we can monitor. So I can use that to see we use it for AMR genes, by the way. We use it for AMR gene what's the name? We said we should come to Agbo well and water. Sorry, I'm not coming to Agbo. So you will discover there are a lot of places in Ibadan, by the way, that I do analysis for. When we get the water and we sequence, you will see a lot of AMR genes, different AMR genes from their wastewater, and you'll be surprised. That means these people here are actually abusing drugs, right? So I think there is a talk about a comparative study of, do we know if humans are the one abusing the drugs most or the veterinary people are the one abusing the drugs? So they want to do a comparative study of, they will sample environmental places where they don't actually rear animals that much, and then they'll go to abattoir and go and sample wastewater from abattoir and compare the rate of AMR analysis, the AMR genes that are found in both of their wastewaters. You can imagine how this would help. So if we know that, okay, these veterinary people are the one actually getting a lot of AMR genes, then we can go ahead and make policies. This is a part of preventative medicine I'm talking about. Make policies that will limit the use of antibiotics for those people because they use it a lot. They use azithromycin, they use ciprofloxacin, they use a lot of fluoroquinolones, by the way, aminoglycosides, different drugs, and you see how they abuse it on their organisms because they want their plants to grow and whatever. So what we are doing here as bioinformaticians is for us to be able to inform decision making so that we can help save the world, make the world a better place. So gene therapy, I have talked about, I won't talk about it again. I talked about gene therapy in my whole conversation. Mathematics sales analysis of genomic data, design gene therapy pipelines, and predict the outcomes and identify mutations causing disease, understand their functions, and develop targets. By integrating bioinformatics with gene editing technologies, researchers can relate and create more precise and effective gene therapies. I mentioned this already, so I wouldn't have to bore us with the whole story and shenanigans again, right? So, how much biology should the bioinformatician know? You need you collaborate with computational people. So bioinformatics and computational biologists develop and test new biological hypotheses, not just analyze data for others. So if biologists think, okay, I think my organism is doing this because of this. How do I validate? Then you as a scientist, as a bioinformatician, we now come with the person, okay, let's formulate this question very well. What data will I need to answer this question? Where will I get the data? How do I come about getting data in an unbiased way? So you need as much biological knowledge as computational knowledge to be able to actually function well. If you just operate tools, that makes you dogmatic. But I think you need quite a number. You need a substantial amount of biology to be able to function well as a bioinformatician. Because there are some questions. Imagine you looking for AMR gene for a drug that is intrinsically resistant to antibiotics. You might not understand what I'm saying now, but don't worry, over time you will know it. So it is you need to have a background knowledge of some biology. So even if you don't know, if you have a question, like this leads me here. If you have a question you want to answer and you don't know the biology, the first thing you should do is go back and read the biology of the organism you want to work with, right? Then understand it, then you can now go ahead from there. Because if you just dabble into it, you might just be merry-go-rounding and be wasting your time. Does it make sense to you? So for every question you want to answer, go back first and understand and read the biology, understand it, then start answering the question. Then you move on from there to the unknown, right? Go from the known to the unknown. So bioinformatician skill sets, biology and medicine, computational science, you will learn these ones, right? Bioinformatics, biological sequence analysis, biological databases, analysis of gene expressions, modeling, then mathematics and statistics, calculus, probability, and linear algebra. So where would you be in this triangle, right? Yeah. Ah. Hmm. Okay. You will learn quite a lot of a number of tools, right? Tools, and more tools. You would use a lot of tools if you want to align, you want to assemble genomes, you want to variant call, you will learn a lot of tools. You would see. And most of the tools are actually open source, by the way, so you might not have problem. Most of the tools are open source. Most of the tools are free, right? And I think we should be releasing this that you can see all of these things. So it is very, very a tool-based work. That's why sometimes I think I enjoy working on servers and clusters because if my computer is just 256 gigabyte RAM, 8 gig, it's an M1 MacBook. So there is just as much whatever I can install. So if you work on clusters, work on servers, you have enough data, enough of space, enough of RAM, then you can install as many tools as you need. You have workflows to install. Very, very beautiful things that you would have to start. You will learn, and by the way, you learn a lot of things. One thing that makes a good bioinformatician is understanding the technical background of the tool they are learning, of they are using, right? So how does this tool work? If I'm using Blast, how does this Blast algorithm work underneath? So that you would not just take the answer face value and think you have gotten it when you know you could actually get better. So two different tools might work the same on the same data and give you two different results, right? So you're using one tool for aligning, you're using another tool for aligning, or you're using one for variance calling, another one for variance calling, and they give you two different results. And then you wonder, is it not the same data I'm giving the both of you? You have to actually understand what is going on underneath these tools. Then you would be able to do better, right? You should be able, you should learn, by the way, to help you, right? So you use a lot of tools. You use a lot of tools. This periodic table, don't worry, you will get you will get at the end of you would be familiar with quite a number of them. Maybe not everything, because most of these things, some of them are even new to me that I don't know. So I will be honest. But over time, you would learn, you would learn, you would learn, and you'll be able to use these tools, by the way. So tool users and tool makers. Um, Tool users are public health informatics, medical informatics, and bioinformaticians, right? Tool makers are information technology infrastructures, development of algorithms, and development of databases. So that's where I'm saying computational and biological will come into place. There are some people that I met someone who built a tool that is popularly used in bioinformatics, and the person really doesn't understand the biology behind it, right? The person made the tool, by the way, but the person don't actually. So these are kind of people that you collaborate with. So me, I'm a biologist. I want a tool that can help me. We're currently working on a tool for Vibro cholerae. I want a tool that can help me type different Vibro cholerae. The one that is existing is bad or shut down. But he does not know. He has the skill that can build, but he does not understand the biology behind it. So it's my own field. It's my job to explain in detail the genomics of Vibro cholerae to him. Explain. So this is how we collaborate, by the way. Explain the whole genomics of Vibro cholerae to him, explain the biology of it to him, explain all the integrations, what makes Vibro, what makes it unique from other organisms, so that he'll be able to build a tool that works perfectly. This man is very, very good, right? So he said, who are the people building these tools? Computer scientists. Computer scientists build some tools, and you would surprise that biologists even built some tools. So I met some people who, after finishing medicine, after not medicine, but microbiology, 4, 5, 6 years, went back to learn computer science and then combined both technology, both what's the name, both domain expertise to build beautiful tools, by the way. And I have known people who were from computational background, they are computer scientists from background, and then after a while they were interested, they picked interest in biology, and then they came to sciences to come and spend 4 or 5 years. I think Thorsten Seaman came to learn, spent 5 years to understand biology, went for another BSc or something. I don't know his story, but he took 5 years specifically to understand biology. He was very good with computer, with programming and building tools normally before in computer science. So after he had understood this, then he combined both knowledge and started building powerful, powerful tools. One of the biggest tools we use in genome annotation, BRCA and Procter and Bacter, he built them. So this is what I'm talking about. So you collaborate with people. Over time, you might start building your tool yourself. If you see that some tools that are existing does not do the solution or does not give you the solution you want, So, but before you start building tools, look around, because almost any problem you will face, somebody else has faced it before. That's just my advice. So, web tools or programming. This is where mathematicians used to like to bully each other, right? So some people will tell you, if you don't use the command line interface and you like to use the web tool, Galaxy, NCBI, you are not a real mathematician. It's a lie. So I think you would collaborate, you would use the tool. Sometimes you would have to, of course, if you want to even pass through your data, sometimes you can use NCBI on their website, and command line too. You can still do the same thing on command line, but it's easier to do it on the web, mostly on the capital than on the NCBI's website. But there are some tools, most tools that you use for your grandiose experiments, they are basically command line. Over when we start, I will teach you how to use the command line, right? So you should be open to learning command line, and this is fast. This is fast, it's reusable. I would favor the command line over the web user GUI. But I want to tell you that, um sorry, I want to tell you that if you use or prefer the GUI over command line, it is not a sin. You are still good. You are still a mathematician. We all want to get results. If you can get your result with GUI, fine. So we have different tools, by the way, tools that are actually primarily web-based that you can't even find on GUI. So genome browsers, when you are done with your analysis and you want to view them, your databases, they are web-based, so you don't have a chance. You have a choice. You have to go there. Galaxy is a web-based mathematics workflow tool that you use. It's online. Then there are software that they are GUI by default. IGV, I use IGV, Patek, Mega, R Studio, people that write R codes. These are mathematics tools that you would have to actually, by fire by force, you have to use web based for that, web based. And then the core bioinformatics, you write bash script, you use by Python, you use Python script, you write R. Yeah, you do data analysis, softwares and different tools that helps you with data analysis are command line based, and then some next generation sequencing tools are actually on the command line too, right? So let me wrap up. I think I've said a lot of repeated things and I've bored us enough. If you are sleeping, you should be awake by now. So in the coming weeks, we'll walk through many of these steps. Our focus will be mainly on genomics. I mentioned it earlier, and I would like you to enjoy the process. You would come through a lot of gates when you start installing tools and they don't work. I said that's the first part where you will be frustrated, but you should soldier on and please move on. You don't have to stress. You would understand it. Don't worry. So get involved in the process, enjoy it, be open-minded, practice as much as possible. I said this earlier, practice as much as possible, and we'll try as much as possible to give you the best of us. So as much as possible, we'll give you the best of what we can deliver. So you don't have to worry about stressing. The best support to you will help you. Just avail yourself and learn, please. Thank you very much for coming to my TED Talk. Yeah, if you have questions and answers. Oh, you Shadula, thank you. If you have questions and answers, please. I have questions and I'll be here to answer. I will answer. Oh, but thank you. Tochi and Ahmed will answer. Thank you, guys. I'm grateful. Sorry I took a lot of your time, by the way. I like to just. Tochi, can you round up and ask? Oh, favor Olua to me, is that? Yeah, please ask. Go ahead, unmute and ask it.
Yes, good evening, Ada.
Good evening.
Thank you for the lecture. Yes, I want to ask a question. So, how
Yeah,
do you translate a wet lab sequencing? How do you translate it into a raw data that people, I mean, they did this thing in the lab. So, how do you get in the sequence? Like, how do you translate into a data that people have to work with?
very beautiful question. So I think that'll be your first lecture next week, from wet lab to data, everything. So what happens is, let's say I have an organism that I want to know the genome of, I want to know what happens in the genome, or I want to know if it has AMR gene or not. What happens is the scientists will culture the organism on the bench. When they culture the organism, they'll make sure it is pure, right? After they are cultured and it is pure, they go ahead to extract DNA. right? There are kits that you can use to extract DNA, quite a number of them. After I've extracted the DNA will be, it's like a fluid, it's a liquid. You would see it in a liquid, right? There are some kits that will make you see the DNA white floating in it, but most of the time, DNA is clear. And then from there, you do something they call library preparation. So sequencing technologies, what they do is they will read your DNA and they will tell you if it is A, T, G, C, and whatever it is, right? So it's after the library preparation, the DNA you have extracted, you create a library for it. You create a library for it, and after you have created the library, you go ahead and sorry, I'm coming. After I created the library, you load it on your sequencer. So your sequencer will then sorry. Your sequencer will now, at the end of the sequencing run, some sequencing run takes it as little as 4 hours. Some sequencing run takes as much as a day, 24 hours, right? It depends. If you are sequencing a small bacteria, a small virus, maybe 2, 3, 4 hours. Then if you are sequencing a human genome, of course, it might take 24 to 36 hours. But at the end of the sequencing, what you get is fast. It's called something called fast queue. You see the data. The data would see ATG. You see that master that I saw ATG, TTCCC. I think I showed that slide earlier. So what you will get is a fast queue, and fast queue, they are not like this, but this is FastQ. So FastQ will have this, and they will have something added to it, like quality score. So that FastQ is what I will now use to do my analysis downstream. So this is how it is gotten. They will sequence the DNA, then the machine will now give you the data. Like it will give you data in gigabytes. You have to use your flash drive to copy it out. So from wet lab, it will convert it to ATGTCCTT, whatever you have, and that's how it works. Do I answer your question?
Yes, thank you.
All right. So, Emmanuel, Miracle.
Thank you very much for the section. It was very insightful. I just
Thank
wanted to know the major difference between bioinformatics and computational biology because sometimes most people confuse the two. So I really want you to expatiate more on the difference
I think
between
it's
both.
a subset. I think it's a a subset of computational biology. I don't think it is different. So it is a subset of computational biology. Computational biology is like a broad umbrella that has a lot of other things that brings. You know, when I was explaining to you before that bioinformatics is not mathematical modeling. So some people do mathematical modeling of healthcare systems. So they are not bioinformatician. They are computational biologists. Do you get? Can you hear me?
Yes, I can hear you.
So they do computational biology, they do mathematical modeling for health systems and whatever. They are all under computational biology together, but they are not bioinformaticians. It's when you start working with genomic data that then you are now a bioinformatician. Do we understand?
Okay. Thank you
Abi,
very much.
yes, you're welcome. Abi wants to add to that. Abi,
Yeah,
before you
so
more.
just to add to that, actually, so I mean, they are two terms that people use interchangeably.
Yeah.
So bioinformatics, system biology, computational biology. So they are all of that, I mean, genomic data sciences. So, I mean, all of them. So, I mean, the umbrella term for everything is just genomic data science. So, actually, so bioinformaticians, I mean, computational biology. So, I mean, if you can go back to that your slide when you were talking about, I mean, the branches of bioinformatics. So, actually, like bioinformaticians, so, okay, let me put it to computational biologists. So they are the ones that actually probably write the algorithm. I mean, they're the one. I mean, that's probably the I mean, design the database, all those things that we are talking about. So, I mean, they develop the tools for bioinformaticians to use. So, in a way, so what we do, so what we'll be teaching you here is, I mean, how to do genome annotation. So, we are not really developing the tools. So, we won't be teaching you how to, like, I mean, we'll be teaching you software development. So we'll just be teaching you, I mean, how to use the tools, I mean, that were developed by the computational biologists. But in this case, I don't think you need to be bothered about terms. I mean, some people call themselves computational biologists. Some people call themselves bioinformaticians. So they are the two, I mean, two terms that you can actually use interchangeably. So in fact, if you actually ask some people, they will even define it the other way and say that, okay, bioinformaticians are the one that develop the algorithm and computational biologists are the one that use the, I mean, algorithm, I mean, developed by the bioinformaticians. So, I mean, there are the two, I mean, there are two words that we use interchangeably, but you don't need to bother yourself with that. So in our own case, we're using the two together. So I mean, consider yourself a computational biologist, consider yourself a bioinformatician. So, but yeah, we won't be teaching you system biology. That's all.
Thank you very much. I asked because when I was filling the form for the training, there was a section where we asked to differentiate between bioinformatics and computational biology. So that's why I'm trying to get the difference between the two.
Yeah, so we just wanted to know your opinion actually, because like I said, people mix them together. So there are some bioinformaticians that they call themselves, I mean, computational biologists. So I mean, they are just two words that people use interchangeably. But you are both in this course.
Thank you.
Okay, maybe just to add an example. Earlier on, Toby mentioned something about AlphaFold from Google. So that's like a good example of what computational biologists do, like trying to model biological systems to do predictive analytics. So that's what they do. Why bioinformaticians like kind of basically focus on analyzing data sets to, give reports and make discoveries. But for computational biologists, it's mostly about modeling biological systems, proteins, amino acids, and the likes to predict one or two things, just like the protein structures that AlphaFold does. Thank you. Okay, Imo, you can go ahead.
Oh, okay. Um, Hello, good evening, everybody. Yeah, sorry, can you hear me?
We can hear you. Can hear you.
Okay. So, thank you. Well done. Thank you for the presentation. I wanted to please clarify something. So, I didn't quite get the application of bioinformatics in drug discovery. Um, well, probably I'm quite curious because um, I studied pharmacy and I think I really want to understand the explanation. I think I understood the application in agriculture, the example that you used regarding the ones. But when you try to explain the application in drug discovery, I didn't understand how. Like, is is it that the um is it does it have to do with the target site of the drug? Like, what exactly in the drug discovery pipeline, where exactly is it applicable? Then you try to give an example on, I think you were giving an example on one MDR-TB um, case or something. I didn't get that example. I don't think I understood it. Most likely. Part. Thank you.
Abib, please can you help me answer? Cooked up something.
Yeah, yes. So thank you very much for that question. So I'm not sure whether you're familiar with this term called, I mean, docking. So I mean, these things that they do a lot in, I mean, I think farm chem and probably some other departments. So okay, imagine having a compound. So you guys work with compound a lot, and then like let's say that probably you have like 10 compounds that you want to screen for activity. 10 compounds, and then let's say probably you have like you have, I mean, you want to test the activities against, let's say probably Klebsiella species. So you want to test it. So normally what you would do is that, okay, we just, I mean, screen. So let's just imagine probably you have like 10 compounds and then you want to screen against, let's say 100 organisms. So if you are doing that, so I mean, it's going to like take a longer time to actually do that. But what you can do is, okay, so can this probably can just goes to the NCBI, download the genome sequence, I mean the genome of all of these isolates. Then use AlphaFold like mentioned, just to narrow down. Okay, before I even start wasting resources in the lab, can I do this thing in silico just to be able to like see whether I will even, because at the end of the day, you want your compounds to bind to something, I mean, on your organism, because you need an interaction for there for something to actually inhibit probably the bacteria. So you need an interaction. So that's actually what you're trying to like do within silico I mean, chemoinformatics or docking. So you are getting the sequence of the isolates, getting their protein sequences, then using like probably docking just to see whether okay, based on the three-dimensional structure of this protein, with my three-dimensional structure of my compound that I want to screen, will it even have any binding effects? So by doing that, you're actually narrowing down probably the number of organisms to actually test your compound against at the end of the day. So instead of probably starting big and then wasting resources, only to say, okay, this drug will not even have any effect at all. Because you would have wasted a lot of resources, time and everything. But within silico, you can just probably use it to narrow down. So these are things that people actually use a lot. Another application is probably if you want to like actually probably determine the target site also, you can actually use probably bioinformatics to actually narrow down also. In fact, so there's somebody here on the code that I'm actually trying to probably assist in doing that. So you can just probably do so. We'll actually teach you when we get to this part. You can do pan-genome analysis. So just to extract the core genes. From the core genes, you shall know that okay, from the core genes, you don't want normally you don't if you are screening for a drug that once you want human to use, you don't want that drug to have any affinity or to actually probably bind to probably any protein or anything in your any cell in your body. So actually, so you can just probably narrow down, okay, I want to actually probably determine the target site of this particular drug. You can actually use in silico approach to actually do that, even without even going to the lab and wasting any resources. You can also use probably, bioinformatics to determine the mechanism of action. We have somebody in our lab that actually did that. As his PhD thesis. Now a lot of companies are looking for him because he was able to like get the target sites, and then now they want to like scale it up. In fact, just that like unfortunately, he doesn't want to like pursue his postdoc, his PhD thesis anymore. So it's not doing like all this NGO. He's more interested in NGO, so he left academics. But there were lots of companies, pharmaceutical companies that actually approached him after his PhD, and that was because he was able to like determine the target site of a drug of a molecule that nobody has even probably tested on bacteria before. So, and then was able to like determine also, the mechanism of action, and he was able to like validate it in the lab. So it was so, by doing bioinformatics, you're actually working smart because at the end of the day, you don't have to like start big and then start wasting resources, but you can probably just use in silico approach to narrow things down so that you can actually probably target your experiment very well. So at the end of the day, you are starting very small, and then so I think probably that is some areas where the bioinformatics can actually come in drug discovery. So there are lots of, I have somebody here on this code that's actually doing something similar for uh, as part of our undergraduate thesis. Yeah, I'm not sure whether you understand.
Oh, okay, thank you very much. It's actually very clear now. I actually get the picture there. Thank you.
Okay,
Sorry,
Favor, do you still want to ask a question? Your hand is still up.
sorry, I just wanted to ask to be if you could please shed more light on the example that he mentioned, saying something about TB, MDR, I don't know, I missed that. I think.
Okay, this is what happened. Sorry, I'm in the middle of like two things together. So this is what happened. I think they used some antibiotics on the patient, and they discovered that in the lab here, and they discovered that it did not work, right? But they have not exhausted all the antibiotics, by the way. So, and we don't have all of them. They just say number of antibiotics you can in the hospital in the lab that we have to test maybe power. So they send the sample to the lab, the research lab, and the they sequence the sample and then run the sample on the database of AMR genes. So they are looking out for AMR genes that are not present in the DNA. Does it make sense to you? So they have DNAs, like they have a database and they see like, of course, the drugs that we have tested in the laboratory that were resistant, we saw their genes in the sample, but they were now under there were genes that we did not find. So we didn't find gene for some organisms from some antibiotics. genes that could cause resistance to antibiotics. So you would infer that the patient can now take the antibiotics. Does it make sense to you? So if the gene is not there in the DNA, then you can give the patient the gene that codes for resistance. Does it make sense to you?
I I still get people. It's fine. You can
Sorry, more like
the
just to understand.
noise background, and I was actually struggling to get.
Yeah, just to understand, I can actually send you like a paper that somebody presented in our some months ago. So probably you can just read it on how, I mean, you can actually use genomics for, I mean, precision medicine, I mean, for clinical treatments. So, I mean, it's a very short paper, just probably 2 pages. And although the patients later died, but what the insight you can actually derive from the paper is, I mean, they were learning, they were just trying to like reflect that what could they have done better if they had access to genomics, and then, I mean, they have computational skills. And then what they did eventually, what they, I mean, they learned from, I mean, the analysis is that, okay, if they had genomics and bioinformatics capacity, they would have actually probably treated the patient very well before he actually probably, I mean, succumb to that sickness. I mean, so I actually, because I mean, in UCH, they're actually using, I mean, they have genomic sequences, so actually using it as part of, I mean, patient treatments, because at the end of the day, so like very soon, instead of probably just using antibiotics, what we'll be using is just going to probably precision medicine and everything. So they will look at, I mean, just sequence everything, and then, so that's, I will send you the paper, I will find the paper on the Google Classroom, and then I'll send it to you. It's a very short paper, just two, 2 pages.
Okay,
Then,
thank
yeah,
you
the
very much, sir.
yeah.
Okay,
So
Favor,
yeah,
your hand is still up. This wants to ask a question. I guess not. So, Richard, I saw your hand earlier on. Do you want to make a comment or ask a question?
Yeah, good afternoon. Good evening. I wanted to say more about the difference between computational biology and bioinformatics, but it seems the person is clear now. So,
Okay, yeah, thank you. So, any more questions, or we should wrap it up? Okay, I guess not. Okay, so please, I have an announcement to make. So please, whenever we send out the link that's to join the meeting, please try and as much as possible to add it to your calendar, because like I've been seeing people trying to join the call, and I'm sure that they are not part of the people that were admitted. So I'm kind of confused. So that I have to like I have to like kind of kick some people out. That's while the call was going on. So please, once you get any mail to join a class, maybe for the next year or thereabouts, try to join, or try to like kind of add it to your calendar rather before maybe the particular time for the meeting, please. Yeah, it will go a long way to help us. Yeah, so
So,
thank you very much. And then one more thing about the WhatsApp. So please, if the number you used to register for the test, or maybe the previous one, yeah, if it's not a WhatsApp number, please kindly email a WhatsApp number to me as soon as possible. Thank you.
sorry, Touch, before you end the call, so I just sent something for Imo and probably other people that are interested in reading this paper. So I sent something to the chat now. So, yeah. So the title of the paper is Detection of Feeding Antibiotic Resistance Through Written Genomics. So you can just actually probably just read it. Say, yeah, it's not clickable. So just copy
copy.
everything and put it on Google. Or you can just copy I mean, that topic alone, detection, I mean, this particular part. So you can just copy it and put it on Google. Then, I mean, it's going to like probably take you to a PubMed article. So yeah, where you can actually read it.
Can you paste it on the group chat on WhatsApp?
But not all of them are on the group chat on WhatsApp. So this one is saying it's not copying. I can copy it now.
Don't worry, I'll email it to everybody.
Okay.
Thank you.
Okay. So yeah.
So I guess that's it. So see you guys next week Friday. Please like try to like kind of attend that class. It's very important. And Angela is coming, so like let's just try to be there. So it will be nice for her to just join and then see a few people on the call. Let's just try to attend on time. Maybe 5 minutes before the time we should all be there to join the call. So I think that's all for now. Thank you very much. And see you guys next week Friday. Thank you very much, Toby, for the class. Good night.
Thanks.
Thank you.
Thank you, sir.
Thank you so much.
Thank you, sir.